Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Ana Cvejic from Biotech Research and Innovation Centre at the University of Copenhagen on this show. Please let me briefly introduce you to our audience. You got your PhD in biochemistry from the University of Bristol in 2008. You then moved on to do a postdoc at the University of Cambridge and started your own lab in 2012 in the Department of Hematology at the University of Cambridge. And since 2022, you are professor at the Biotechnology and Innovation Research Centre at the University of Copenhagen. A question I like to ask to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
So I think that I made a decision to be scientist very early in my life. I was probably 11 or 12 years old, and I think it was kind of linked with. Obviously, it was such a long time ago that I don't know any details, but I think it's probably linked with what we started studying in biology, and I think probably initially it was mostly linked with the. Maybe fascination with viruses and bacteria, nothing that I work now on. But that just probably triggered the interest. So it was very clear very early on in my mind that this is what I want to do. Maybe a little bit of an issue was that I was not, so my parents didn't even have a university degree. So I was actually the first person to get the university degree, not only in my family, but kind of even in a broader family. So more of a struggle was how to find the way, how to achieve what I knew that I wanted. There was no one really to guide me or advise me and so on. So my parents were very supportive in a sense, you should do what you would like to do, but they were not able to help me navigate the way to get from where I was to get a university degree and then PhD and then after that postdoc, and so they didn't know much about that. Obviously, the country that I was born in at the time was not maybe the best place to do science, so that was a struggle as well. But that obviously came later, and realization that I would need to leave came later on. But the initial decision happened very early in my life.
Yeah, it's interesting because many of the people that have been on the podcast, they kind of stumbled into it. They didn't really know where to go, but it's very interesting that you had this in mind very early on.
And I was very clear.
Did you then realize what it meant to go with this path? So, because you knew what you wanted to do, but I mean, at least for me, I knew that, I want to do science, but I mean, doing all the studies and then a PhD is a very different thing than just doing science, right?
So I think that the whole path in terms of educational part of becoming a scientist, meaning going to university and get a PhD, I didn't find that problematic. I think I found a more problematic part when I became group leader. And obviously at that time, I was not aware what it means what it means or what you need to do actually to have a successful lab. How difficult it is to actually even reach to the place where you can become a group leader. So that is the transition from being a postdoc to being a group leader and then later on to get a professorship. And I didn't know that obviously at that time. And it's good that I didn't know, because I think it would be very discouraging if I did know. So I think for me, I was very determined. It was very clear in my head what I want to do. And I was taking these obstacles one at a time, so to say, and then trying to solve this problem as they were coming, and then got to but now looking back, obviously, it's a long path and it's a difficult one. So I told my children they're not going to do that.
So do you think that it's important to have this kind of mindset to be successful in science?
I think so. Yeah, I think these days, yes. I think that the face of science and the way how you do science and the way how you get in a position to be able to do science in a kind of independent way. These days, I think you do have to have a certain mindset and level of determination and many other kind of features, psychological features. Not only to be smart. That's almost kind of almost becomes irrelevant.
The default, yeah.
Yeah, no, exactly. That's a default. And then you need a lot of other things that are necessary for you to get there, I think.
Yeah, maybe we can come back to that later, because obviously I want to talk about the science you're doing today. And this centers around using single-cell methodologies to understand hematopoiesis and immunology. I want to start, as we always do in the beginning. When you started your own lab, the first paper published was a meta-analysis with the title "A Loss of Function Screen of Identified Genome-Wide Association Study Loci Reveals New Genes Controlling Hematopoiesis." Can you talk about how you got into the field of hematopoiesis now more specifically, and what you learned from the study?
Right. So I think in all of the work that I did, there is always a little bit of background in how I got to a certain position. And so the first paper that came out of my lab was in a way continuation of the work that I did during my postdoc. So during my postdoc, I joined a group of Willem Ochen. So I was in a way jointly supervised between him and Derek Stempel. And it was a nice combination because Willem was interested in genome-wide association studies and identifying novel genes for regulation of blood traits like red blood cell count, platelet count, or size, and so on. Whereas Derek was running a group that was, he was also at some point ahead of mouse and zebrafish genetics program at the Sanger Institute. And really the idea was that we use a zebrafish as a model system to validate some of these newly discovered genes. And this is where my expertise that I got through my PhD came handy, because I had a lot of expertise with using zebrafish as a model system, but maybe mostly in the context of wound healing rather than hematopoiesis, whereas Willem was kind of more interested in mostly actually platelets, but during my postdoc I think that there were also his kind of interest expanded. So that got me a little bit into field. And the way I was thinking during my postdoc is because this was such a new field and very interesting one, I managed to publish the nice number of papers that were sufficient for me to start my own group. And when I was thinking about that, I was being very pragmatic, and I was thinking I should probably continue with things that I know really well to be able to set up my lab and publish some papers. And this is how this paper came out. So basically the genes that we were validating in that study were genes that came out of the work when I was a postdoc in Willem's group. So these were many genes that came from a Jiva study. And like as a part of the start of my group, we started validating them. So looking at what is their role, broader role in hematopoiesis. And this is kind of slowly kind of got me in hematopoiesis. So I was moving just from looking at the number of thrombocytes in zebrafish or erythrocytes, kind of looking at the broader effect of these genes. So this is how I got there. And this is what that paper was about. So it's really about validation of the novel genes.
Yeah. So in the title it says loci, and you now talk about genes. So this was only about the coding part of the genome, or were you also looking at like regulatory elements?
No, so this is very early days. So yeah, you're right. It is loci, and I'm talking about genes. So basically, what we had to do is we connect these loci with the genes in a way, like in a simple way. And I mean that's why it had to be a screen, because there was no guarantee, obviously, that this gene was kind of regulated by that SNP, and obviously it was kind of. With the SNPs, you're looking at the changes in a level of gene expression. Here, I was knocking down genes. So it's a little bit different. You are kind of almost taking the phenotype in extreme. You're thinking, okay, I'm going to substantially decrease the level of something, a particular gene, and then ask a question: what is their role in hematopoiesis? And obviously, the benefit of zebrafish was to be able to do a larger number of genes, but the limitation was also that you're doing that within a kind of narrow period of zebrafish development, which means that we were mostly looking at developmental hematopoiesis, not what was happening in adult zebrafish. So there are many limitations basically in this study, which then brought me to kind of move away from that to next part, which is linked with single cell
Yeah, exactly. Let's dive right into that. You then moved on and turned to single-cell studies, and you applied a single-cell RNA sequencing to a population of hematopoietic cells in zebrafish, as you already mentioned, as they undergo thrombocyte lineage commitment, and you were able to construct a continuum from stem cell to a mature cell using this approach, which is very nice thing to do. You just wanted to go in. I just wanted to set the stage and.
So I'll kind of further set the stage. This is to kind of explain what was going on at that time. So at that time, I'm doing these morpholino knockdowns in zebrafish, and I'm being a little bit frustrated by the fact that I didn't feel that I was going deep enough with the analysis. Obviously, one can then after a screen, you can go and do a more kind of in-depth functional analysis of individual genes, but at the same time, at that point of time, I was not familiar with and there is a reason for that. I was not familiar with the kind of anything that had to do with the single cell technologies, and the reason was obviously because they were just emerging, right? And so what was happening at the Sanger at that time is that they were trying to set up these technologies, and obviously through various seminars and by the very fact that I was there, I was aware of these efforts. And then I came to an idea that actually maybe it would be good to use the single cell technologies to try and reconstruct the lineage tree. So to try and kind of look at the process of differentiation by using single cell technologies. And at that time there was a paper that came out where they actually followed in cell culture, in vitro, differentiation of cells. And they were collecting them at different time points, and they kind of had tool to reconstruct changes in gene expression. And my idea at that time was, okay, why don't we do that in vivo? And I had a model, so I had zebrafish, and I was familiar with thrombocytes. I had all the tools that I needed, except single cell technology. So then I thought, okay, we could maybe try this. And at the same time, I was collaborating with another group at the Sanger Institute to try and set up the experimental part, but also to try and analyze the data. And at the same time, I made a strategic decision that this is going to be the direction in which I'm going to go. And if I am going to go there, then I'm going to need money. And therefore, I decided to apply for the ERC starting grant. So this is how all of this is happening. So at the same time, we're doing this study and testing, actually, because some people told me it won't work. Trying to kind of see, can we take a broad marker and capture the whole continuity? And at the same time, trying to get funding to extend that line of work in my lab and kind of make it as some sort of direction. And the bonus on top of that was the fact that I felt that this is something that is my own, meaning that I was stepping away from my postdoc work, and I was taking ownership of a piece of work that I was kind of leading on and in charge of, and so on. So that was very interesting. And now, compared to all other studies like came after with these really small studies, so we maybe had like 300 cells, something like that. So, but this is 2015, so early days. And it was very nice because we could actually show that there is this continuity in the differentiation, and we could see that actually there is some coordination in the transcriptional programs which are happening during this the process of differentiation. And in that case, we were looking at the thrombocytic lineage, and it was kind of increased with the suppression of genes which are involved in cell proliferation. So as cells are moving from stem cells toward mature, they're kind of suppressing these, and ribosomal biogenesis, whereas the genes which are important for the lineage differentiation were going. So that was some sort of main findings that we had from that study.
So this study focused on single-cell RNA-seq, and I think, I can't remember the number correctly, but it said 600 cells, I think.
I think it was 350 or something like that. Very small number of cells. Yeah.
So the obvious thing to do is going from RNA-seq and including ATAC-seq, obviously, and then also use more cells than that. So what you did is combine single-cell RNA-seq with single-cell ATAC-seq and use 8,000 cells. And you call this then immunophenotypic. Maybe you can just go into the details what that means exactly, and what you learned from this study, then expanding everything to more methods, to more cells, and about the relationship between chromatin accessibility and RNA expression or gene expression.
But there is, you kind of skipped some steps there. So I'm going to again give you a background there. So the studies that I was talking initially were done in zebrafish, right? And the study that you are now mentioning, that's actually done in human. And
I see.
Yes. So I had to make a big step between, again, for me, to make a strategic decision to move away from zebrafish to actually go into the human. And as I always kind of make my decisions, it's kind of linked with some level of frustration about the how deep I can go with a certain model, and maybe to some extent how easy or difficult it is to get the funding using a certain model. So I think initially, using zebrafish as a model system was excellent to start with. But at some point, I felt I wanted to do more, and I wanted to move to kind of more relevant model. I wanted to actually move into human. And so that study that you're describing, which we actually published in Cell Stem Cell in 2021, that's very kind of close to my heart, because it's the first one where it took us a couple of years actually to make this transition to be able to get human fetal samples. So this is developmental hematopoiesis, but from the really kind of looking in human, and questioning some of the kind of traditional ways how cells are being sorted and analyzed in that field. And so this immunophenotypic that you are mentioning, it's referring to the kind of traditional way of defining stem cells and progenitor cells and mature cells by a set of well-defined surface markers or antibodies. So you have an antibody panel, and then you sort these cells, and then that's what defines the identity of the cell.
Sorry to interrupt, but so this was not only about answering a biological question, but also like finding new methods to do the things to answer those biological questions?
But these things are always linked, because you're in a way restricted by the technology, right? And how people are thinking about the science. That, when you get a result, that result should be always considered in the context of the technology that you used, right? So people were mostly using these cell surface markers to define the identity of the cell. And really the idea here was they were twofold. So first of all, can we better describe the identity of cell if we were looking at the thousands of genes rather than a handful of cell surface markers? And secondly, is it true really that these markers, which have been shown to be kind of relevant in adult hematopoiesis, is this really true during developmental one? So can we actually, and maybe can we define other markers? And markers are always important, because at the end of the day, you want to sort these cells and you want to functionally do something with them. So you have to have the information about how to enrich for your population. But the question is, are we aware how heterogeneous is our population when we do something like that? And then again, study was quite nice, and we got really nice results in a sense that actually what we were able to define are new cell surface markers to better enrich for the cell populations during fetal development. Secondly, that the classically used markers are actually collecting heterogeneous population of cells. So this was on a technical element. And then more on a kind of biological element, we were able to show, a biological side, we were able to show that, and we were focusing here on hematopoietic stem cells, is that because we had information from the ATAC-seq, so chromatin accessibility and gene expression, we could actually ask the question, how are the cell fate choices made? So there is a big big part of the field is so-called priming of hematopoietic stem cells, when they are not committed to go somewhere, but they have a tendency or bias to go somewhere. And so we were able to actually show that this priming in a way starts on a level of the chromatin accessibility, and then it's followed by gene expression. And that was quite nice, and actually made the basis for some of the work that we are doing now. Not yet published work, but something that we are now doing.
So what is the time scale there if you are talking about priming and then lineage commitment and gene expression. So can you give some ballpark numbers here?
No, no. So I think it's at some point it actually becomes a little bit philosophical. So in terms of the real time, not really, but it's just conceptually, it would be a situation in which a stem cell has a potential to go to many different places. Then you can have priming, where you have this disconnect between chromatin accessibility and gene expression, meaning that on a chromatin level, there is maybe it's primed to go in a certain direction, but genes are still not being expressed. And then you will have a commitment, and at that point, it's still reversible. So I think that's an important point, that it still has, it's primed to go a certain way, but it has still ability to maintain its stemness and go in many different ways, depending maybe of external signaling. And then there is commitment where you actually are committed and there is no going back. You're going in a certain direction. Now, time-wise, how long that lasts, I mean, that's an interesting question. Maybe someone knows the answer, but I don't.
And I guess I can imagine what your current work would be about, because obviously the next question would be, what are the factors that are involved in this processes, right? What makes it irreversible? When is it irreversible? Is that what you're currently focusing on?
So I think what we are currently want to publish and in that sense focusing on is this more kind of going on a question of priming and how is that kind of linked. So in a way trying to kind of get the full picture what that priming actually means. And the model that in a way on which we are working is maybe model is not the best way, but a condition in which we're looking is a Down syndrome. So it's a trisomy of the chromosome 21. And the reason why we chose that one is that it is well known that the children with Down syndrome have higher probability of developing leukemia, but also independent of leukemia development, they also have many of newborns have polycythemia, so they have increase in the number of erythrocytes. So the question then is, can we see at the level of the stem cell this kind of priming which is going to push their differentiation toward erythroid lineage? And this is then where, in a way, looking at the chromatin accessibility and the gene expression becomes very handy, because there are tools in which you can link these two and see actually which of the kind of open regions are linked with which genes and how they're changing in the context of trisomy. But a very important point, unlike the paper that we were discussing before, here this chromatin accessibility and gene expression are coupled. So we are getting information for each cell about two modalities. That is very different from what the previous study in which we had chromatin accessibility from one population of the cells and the gene expression from the same population, but not the same cell. So these two modalities were kind of uncoupled. And again, out of frustration to link these, we decided to go into this is called Multiome, to have from the same cell chromatin accessibility and gene expression. And then you can actually nicely see how it works really well. It's yeah, right.
But it's not published, so I don't want to go into more details. Last but not least, you reconstructed a phylogenetic tree of blood development using whole genome sequencing of 511 single-cell derived hematopoietic colonies from healthy human fetuses at 8 and 18 weeks after conception. What was the motivation behind this, and what did you find?
So motivation for this, or the reasons for doing this study were coming. So the main idea, basically, or main question, there were two questions that we started with, and then we answered many more, and maybe not the ones that we were initially asking, but it was very nice and elegant kind of study. So we were interested in kind of trying to understand how many stem cells are made in a fetus. So because there is a narrow window within which you make stem cells, and everything after that is expansion of the initial one. So the question was how many in humans there are. And there were some kind of numbers in zebrafish. There is like 30, or in mouse, 600. So the question was, how many in human? And the second was what we were interested in is that the process of blood development in human is happening across multiple sites. And first, they're kind of specified in something which is called aorta gonadomesonephros. Then they move into fetal liver, and then from fetal liver they move to the bone marrow. And bone marrow is a place where blood is made in adults in normal situation. And so we wanted to know if during fetal development cells are going back and forth between two sites, because it's really interesting. In one fetus, you have, in one fetus you have two sites and of hematopoiesis. And so what is happening with these stem cells? So this is what we kind of started with. But we then ended up kind of answering many more questions. So the study, I don't know if you want me to go a bit into the way how the study was designed, because it explains kind of logic. So this is, and I have to kind of highlight this. This was done in collaboration with Peter Campbell. And he previously published already a paper in which he, in a way, showed that we can use somatic mutations to do the lineage tracing in humans. So what that means is that if a stem cell or progenitor cell acquires a mutation, the progeny of that cell will have that mutation, so it's inherited. So in a way, you can follow the lineage. And secondly, if any new mutations are acquired in any of these now progenitors, then their progeny will inherit these mutations. And so if you have now information about all of these mutations, and if you do whole genome sequencing, then you can build phylogenetic tree. So you can build a tree which is telling you about the relationships of different in this case, stem cells and progenitors. And so this is what we did. But the element which was really elegant is that we also collected other non-blood tissues. So we could ask the question, not only, for example, how many mutations are present in human stem cells at 8 weeks post-conception or 18 weeks post-conception. We could actually determine a time, real time in development when these mutations have been acquired. Because if the mutation is shared between blood and another tissue, that means that mutation had to be acquired before these two tissues kind of separated. And so this is the elegant part of the study. So we learned a lot from there. We could actually see that the rate of mutation acquisition is not constant during development. We could see that we could actually give a number how many mutations are acquired in stem cells early in development. We could build this phylogenetic tree, and we could make some sort of, not precise, but vague estimate of how many clones there are. And I think what we are saying is that there were at least 1,000. So at least 1,000 cells were initially made. So that was some of the interesting findings. But I think it was a really collaborative effort. I must say that. So it's not only us. It's Peter's. Yeah.
So we kind of touched upon my next question, but maybe there is more to add to this. So imagine you are due to submitting a grant proposal tomorrow. What would you have written into that?
I am, but I'm not going to tell you what I'm going to write there. I'm going to tell you about something else. I'm just going to globally or generally say that there is a lot in terms of the human developmental hematopoiesis that we don't know about. And it's in a way, it's linked with a kind of functionality of the stem cells during development. And there is a lot of talk that early stem cells are not fully functional, and then they kind of become mature and so on. And understanding how that process is happening and to what extent it's cell intrinsic or cell extrinsic and so on is super interesting. And the other thing, actually, maybe the answer to your question, if I was writing another grant in addition to the one that I'm now, the study where we built a phylogenetic tree of blood, what I think it would be interesting is to build one that would cover all of the human tissues. So basically have a complete phylogenetic tree of human development. Yeah, that would require kind of some sort of consortia, and people with various expertise and a lot of money. But I think it would be worth. We would learn a lot about human development.
So for the last 30 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
You decided to have this podcast, which is on a kind of scientific part, but I do think that the way one does science and how well you do it, and then moving forward, like what the whole of science should look like depends a lot on the individual and the kind of barriers that exist. And some of them we are working on, some of them not that much. Like there is a lot of talk about the gender balance, but there are also other things because quite often scientists would travel from one place to another and move. And so there is a question of diversity and so on. And I think these are important parts, but they're not focus of your podcast. So maybe not.
No. We can go into that if you want to talk about that. That's a part of your story. And this is also what this is about.
Yes. And I think that's a good thing that you said. So it's a part of my story. I do believe it's not only part of I do think it's a part of other, like maybe women's. If you make it about women, like we can do it like that. As I said, there is now maybe more awareness and there is a little bit more work on that. But I think that there is a lot to be done. And I think, you can put some regulations and you can put maybe even quota and trying that way to sort a problem. But I think the problem is much deeper, like, and one can see that in interactions with not all colleagues, but some colleagues when, you feel that you're maybe not taken as seriously as one should be taking you seriously, like in discussion, which is, I always kind of think like, maybe age, gender, where you're coming from, and so on.
Yeah, it's very hard because you also need to be very flexible, right? I mean, you should. I mean, family and scientific career is always hard to combine, right? I mean, it's very hard, even more so for women as for men. But in the end, you at least need to move once or twice in your career, right? Totally move your household and everything. And this is also something that maybe is not for everyone.
It's not for everyone, but I think it's also not. It's not discussed.
Yeah.
And it's not appreciated in a way. So I feel that the system is like university system, or the way of thinking in academia is lagging to what is happening in industry and so on. So I think that there is not a good understanding of. I just feel that the system is not sustainable as it is. Like, I mean, you can see that with a number of postdocs that are leaving academia and going to industry and so on. So I think that the academia will have a problem in the future if it doesn't address the issues now.
Yeah, hopefully there will be some changes made or thought about at least. In the beginning, sorry, go ahead.
Yeah, no, they will have to. Otherwise, one will lose the talent, right?
In the beginning, we were also talking about like personal traits that one needs to have to be in academia to follow this scientific part. Can you maybe also from your own experience tell us what you think would be the ones to have?
Oh yeah. So I mean, obviously, I'm looking from my own perspective, and that is shaped, like you said, by my journey. And so to kind of again put in perspective, I'm a kind of female coming from Eastern Europe, from Serbia. And at the time when that country was not in the best place, like I was actually born in Yugoslavia, and then there were wars and so on. So from the economic point of view, it was a difficult time, and also politically and so on. And so to go from there to, so I mean, to actually even leave, or I can maybe say this is something that I don't like when people are doing, but I was doing that. So when I wanted to do my PhD, I sent 200 emails, very generic ones, in a kind of desperate need to get the PhD position. And for example, nowadays, if someone is sending me generic email, I do not appreciate that. But I understand. I mean, I went through that. I understand where is that coming from. So it's a learning curve. You learn. I mean, first of all, it requires a lot of perseverance. Like, you have to really be determined. You have to sacrifice a lot of things, and you have to learn on your mistakes. That's quite often people are just they're maybe criticizing you, and I don't like being criticized, but I will think about it, and I really I will try to improve kind of my next move or next step based on the advice. And I think that without that, I don't think I would be able to get where I did.
And you have to be very, you have to forget things that I mean, on the one hand, you need to improve on your mistakes that you did. But on the other hand, there are so many things that you can like control, and you need to forget the emotions at least that you felt when you did an experiment and it didn't work. I mean, there are so many failed experiments that, I mean, but again,
there is a lot of failure. There is a lot of failure. I mean, actually, I mean, that is the problem maybe with this line of job. You're criticized the whole time. So when you apply for grants and you get comments from reviewers, they're criticizing you. When you're applying, when you're kind of submitting your work on which you worked like 3 or 4 years or more, and then it's kind of trashed by another anonymous reviewer. So you have to kind of be able to survive all of this criticism and keep going. And there are periods when you have a lot of rejections. You know, you apply for one funding and second one and you're rejected and so on. And then you have periods when you get the funding. So it's just kind of surviving the tough periods to kind of then be able to enjoy the ones when things are going well. So yeah, it is unusual, I would say, profession in that sense. Yeah. That level of criticism the whole time. Yeah.
So that was the last question I had. Do you want to add still something, or?
No, I think that covers it. Yeah.
Okay. So perfect. Thank you, Ana, for your time and for being on the show.
Yeah. Well, thank you for inviting me and spending time with me talking about this.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.